Today's lecture will be about specific immunity. So in specific immunity, immune system cells target specific pathogens. So let's talk about the pathogen. What is a pathogen? A pathogen is a disease-causing agent. Right? So we'll draw a pathogen. And on this pathogen, you'll have surface proteins that identify this pathogen. We call these antigens. So the two types of immune system cells that we're going to discuss are the T cell and the B cell, right? Which are also called lymphocytes. So you have a T lymphocyte and you have a B lymphocyte. Okay? And we'll talk more about these different types of cells in the next discussion. Okay? So when you look at an immune system cell will have a specific shaped receptor. So this right here, this is a receptor. And if you notice, the shape of this receptor resembles the shape of this antigen. Hence, this is why we call it specific immunity. Likewise, with a B cell. A B cell will have a specific shaped receptor for a specific shaped antigen. This lecture is about a type of specific immunity called cell-mediated immunity. And in cell-mediated immunity, the major players are the helper T cell and the cytotoxic T cell. So in cell-mediated immunity, the first thing that has to happen, unfortunately, is that you have to get infected. So here you have a pathogen. And on this pathogen, you have antigens, right? So this is a pathogen. Right? Remember, pathogen is a disease-causing agent. And on the surface of this pathogen, you have antigens. After you've been infected, then you have a specific type of white blood cell called a macrophage. Okay? And this macrophage kind of patrols the area, if you will. And this macrophage is going to eat this pathogen. The cool thing about this macrophage is that after it eats this pathogen, it's going to display the antigens of that pathogen on its surface. And it's going to display these antigens on its surface on something called a major histocompatibility complex. Major histocompatibility complex. In humans, we call this HLA, right? Human leukocyte associated antigen. So when this macrophage eats this pathogen, again, it displays the antigens of that pathogen on its surface on a major histocompatibility complex called the human leukocyte-associated antigen. Once those antigens have been displayed, the first lymphocyte that comes in contact with this macrophage will be this cell right here. This is called a helper T cell. In the helper T cell, right, you can see that it has a specific shaped receptor that'll bind to this specific shaped antigen. So these two things will bind. When that happens, right, the binding of the helper T cell to the antigen stimulates the macrophage to release a cytokine. Right, called interleukin 1. Right. And that cytokine acts upon the helper T cell. And that cytokine stimulates helper T cells to multiply in number, but it also stimulates the helper T cell to release another substance, another cytokine, called interleukin 2. And interleukin 2 attracts other types of lymphocytes. And since we're talking about cell-mediated immunity, the lymphocyte that interleukin 2 is going to attract is a lymphocyte called a cytotoxic T cell. So you can see that this cytotoxic T cell has a specific receptor that fits the shape of that particular antigen. Okay. Interleukin 2 attracts cytotoxic T cells, but it also stimulates cytotoxic T cells to proliferate. Proliferate means to increase numbers. In other words, your body is trying to raise an army to fight a war. So we're going to draw a few cytotoxic T cells. And we are going to multiply these cytotoxic T cells here. Right. We're going to increase their numbers. All right, just like that. Okay. Right. So some of these cytotoxic T cells won't go to battle at this point in time. We call these memory T cells. And these memory T cells will become activated the next time this pathogen enters the body. Then there are these cytotoxic T cells that will go into battle. Right? So these are just cytotoxic T cells. So when a cytotoxic T cell, right, encounters a pathogen, so let's draw the pathogen. Right. That cytotoxic T cell's receptor will bind to that antigen, and this binding will stimulate the cytotoxic T cell to release a substance that'll adhere to the membrane of that pathogen. We call this substance perforin. And as the name implies, perforin is going to perforate or create a hole in that cell membrane. So what happens is, as that cell membrane has a hole, right? So here's the hole to the cell membrane. Fluid is able to rush in, and this causes the cell, the pathogen, to burst. The next type of specific immunity that we're going to discuss is antibody mediated immunity. And the major immune system cells in antibody-mediated immunity, again, are the helper T cell, and this time is a B cell. So just like before in cell-mediated immunity, unfortunately, you have to be infected. So here again, we're drawing a pathogen, and on the surface of this pathogen, we are drawing squared-shaped antigens. So once this pathogen has invaded the body, right, it encounters a macrophage. So again, this is a macrophage. Just in case you forgot, this is a pathogen. And that macrophage, once it eats this pathogen, will display the antigens of that pathogen on its surface, right, on what we call a major histocompatibility complex, and in humans we call it HLA. Okay. Then we have another helper T cell. That encounters this macrophage. And again, this helper T cell has a specific shaped receptor to a specific shaped antigen. And when this helper T cell binds to this antigen, it stimulates this macrophage to release a cytokine called interleukin-1. Interleukin-1 stimulates helper T cells to multiply in number, right, to proliferate, okay, increase in number. But it also stimulates helper T cells to secrete another type of cytokine called interleukin-2. And interleukin-2 is going to attract a new type of lymphocyte. And since we're talking about antibody-mediated immunity, this new lymphocyte that it attracts is called a B cell. And if you notice, this B cell has a specific receptor for that particular antigen. Okay. So interleukin-2 acts on this B cell and stimulates this B cell to multiply in number. Right? You know this as proliferate or multiply in number. Right? So again, we're going to draw a couple B cells. And we're going to have them multiply in number. Alright. So just like before, some of these B cells will not go into battle right away. These guys are what we call memory B cells. And then there are another set of B cells that will be activated. And these B cells will become what we call plasma B cells. Now, plasma B cells are going to make something called an antibody. And antibodies are proteins. Right? So these antibodies are going to be Y-shaped structures. Let me use green. Right? Y-shaped proteins. And at the ends of these antibodies are these antigen binding sites. And if you notice, these antigen binding sites have a specific shape for a specific antigen. So when these antibodies encounter this pathogen with that specific antigen, right, so here's the pathogen, and here's the antigens on its surface. So when these antigens, when these pathogen antibodies, right, encounter this pathogen, these antibodies cling on to this pathogen. So let's try another. Right. And basically these antigens, these antibodies slow the pathogens down. Right? So here are the antibodies. They bound to the antigens of the pathogens, and basically they clump them together. Right? And what that does is it slows these pathogens from doing anything, so that maybe a macrophage, right, can engulf them, or even maybe a cytotoxic T cell can release perforin to puncture a hole through the pathogen's membrane. In this lecture, we're going to talk about HIV, which stands for Human Immunodeficiency Virus. And what makes HIV so deadly for the human immune system is the type of lymphocyte that it attacks. HIV specifically attacks a helper T cell. So in this illustration here, this is HIV. And this guy here is a helper T cell. And on the surface of a helper T cell, you have a receptor. And this receptor is called a CD4 receptor. This is the receptor that a surface molecule on an HIV virus, right, a surface protein on an HIV virus, specifically binds to. So HIV will bind to the CD4 receptor. And within HIV, you have these like little squiggly lines right here. This is viral RNA. HIV is what we call a retrovirus. And it's a retrovirus because it uses RNA as its genetic material as opposed to using DNA. Also inside this HIV virus, this little yellow dot right here. That yellow dot is called reverse transcriptase. So you'll have to kind of remember back in your biology days about a process called transcription and translation, which was using DNA, the blueprint, to produce proteins. Okay? So reverse transcriptase is going to have a role in that process. Okay? So once this HIV virus binds to a CD4 receptor on a helper T cell, the membranes of the virus and the membranes of the helper T cell fuse together. And by doing this, the HIV viral RNA and the reverse transcriptase are released into the cell. So here you have viral RNA and reverse transcriptase being released into the cell. And now what happens is that reverse transcriptase, that reverse transcriptase converts viral RNA to viral DNA. In other words, reverse transcriptase transcribes viral RNA into a viral DNA. And then that viral DNA will migrate itself into the nucleus of a helper T cell. So here you have viral DNA. So this is viral DNA. And that viral DNA will fuse itself into the host cell's DNA, right? And then what happens is transcriptase. So transcription. Probably can't read that. Let's change that up a little bit. Okay. So we have transcription take place. Transcription. Okay. So transcription of this new DNA molecule that includes the viral DNA molecule will produce this thing right here, right? That new thing is what we call viral mRNA. Viral mRNA. And that viral mRNA will be sent to this guy right here. This is a ribosome. A ribosome is an organelle that makes proteins. So this viral mRNA is delivered to this ribosome, and this viral mRNA codes for the message to make these guys here called viral proteins. And those viral proteins will be assembled, and they'll be assembled into a new HIV virus. And when a new HIV virus or the insides of an HIV virus are created, then it buds off, ready to infect another helper T cell. So this here is a new HIV that buds off.